I'm going to talk primarily a little bit about the work that I did while I was still in Carlie's lab. And that is trying to understand how existing oligodendrocytes help modulate neuronal activity and help us learn. And so Ben did a really good job of already explaining what an oligodendrocyte is and describing myelination and the formation of internodes and the nodes of Ranvier. So this configuration is actually really important for saltatory conduction, which helps the rapid and reliable transport of action potentials along myelinated axons. And so this is something that I'm really interested in, is what role myelin has in kind of modulating the speed of that action potential along these axons and kind of the role that plays in learning. So there's a few things that can actually modulate conduction speed in a myelinated axon. The first thing is axon diameter. Obviously, larger axons are more likely to be myelinated, but they also will transport action potentials a lot faster. The other thing is the pattern of myelination. So we know now that not all neurons are myelinated equally, and there's often quite large gaps in between the myelination, and internode length is very different, and there's different variations in the thickness of myelin, even along a single axon. And one of the other things is the node of Ranvier. So the position and the size of the node of Ranvier and the density of the sodium channels that actually cluster there can also help modulate the speed of action potentials, even along a single axon, so it can vary the speed. We know that we can modulate the pattern of myelination through the addition of new oligodendrocytes. So again, Ben and Carlie have already told you that OPCs generate new oligodendrocytes throughout life. And so by the addition of new oligodendrocytes, we can change the pattern of myelination along an axon or within a circuit. And this is just a very small selection of a wealth of literature that's out there showing that if you modulate neuronal activity, you can actually drive this addition of new oligodendrocytes. In addition to exogenous stimulation or artificial stimulation of the neuronal circuit, if we can endogenously activate a neuronal circuit, say through learning, for example, here I'm just showing spatial learning, we can also drive the addition of new oligodendrocytes in brain regions that are associated with that form of learning, like the retrosplenial cortex, CA1. So all of these regions show an increase in oligodendrocytes after Morris Watermay's training spatial learning. But what's really interesting is that despite years of us thinking that once myelin is laid down and an oligodendrocyte is established, the length of its internode, it's quite static and it just sits there until the oligodendrocyte dies. We now know that that's not true, and that the existing internodes can be dynamic, and so the oligodendrocyte can adapt to changes in the environment. So the video that's repeatedly playing here is in a developing zebrafish spinal cord, where Tim Chopka has actually ablated a single oligodendrocyte to remove a internode along an axon. And you can see that the established internodes either side actually kind of encroach on the space to fill the gap to presumably accommodate for the change in conduction that's occurring. That doesn't just happen in the developing nervous system. Robbie Hill actually showed that this continues to occur in the mouse brain as well. So these established internodes, the myelin internodes, or established oligodendrocytes, can actually extend or retract internodes over time. So this led me to the question, can existing oligodendrocytes remodel during learning? And so in order to look at this, we used a transgenic mouse approach, so Krylov transgenesis, to fate map existing oligodendrocytes. So again, Ben already introduced the PLP-Cre model, and we actually crossed this line with a tau and GFP, which is a membrane-targeted GFP under the tau promoter. And so what happens is when we give tamoxifen at a certain age, so we've given it at P60 or 8 weeks of age, we can label a subset of existing oligodendrocytes. So these are the cells that express PLP, so they've already been established. So the majority of these oligodendrocytes would have been born developmentally at this stage. We then put these mice through a spatial learning task. We chose the radial arm maze task, in which mice actually have to learn to retrieve a little Fruit Loop treat from each arm and learn that once they've entered the arm and eaten the treat, they actually don't get another one if they repeat, if they re-enter the arm. And so this is just showing that over the 11 days of training, they actually improve and make less errors over time. So they re-enter the arm that they've already entered less over time. So we then collected the brains from these animals and we had a look at the fimbria, the fimbriafornix pathway, so the hippocampal fimbria. So spatial learning, or the radial arm maze task, is very hippocampal dependent. So we decided to look in the white matter tract, a major white matter tract that projects from the hippocampus through to various cerebral cortex regions and the prelimbic cortex and the full brain, sorry. What we actually found though was that in our learning mice, so our no learning mice got exposed to the maze, sorry, I should have said this already, but they got exposed to the maze, but and to the Fruit Loops, but they never had to navigate the maze. So they didn't actually learn, whereas our learning mice had to learn to retrieve their treats. What we actually found when we looked at internode length after learning was that there was absolutely no difference in the length of internodes in the learning mice and the hippocampal fimbria versus the mice that didn't learn. And so this was somewhat disappointing and probably should have ended there, but I'm stubborn. So I wanted to look at the nodes of Ranvier. And so this is what we did here, just to show you what's going on here. So we've got the paranode labeled conduction associated protein labeled in green, which is the paranode, which is where the myelin tethers to the axon, and sodium channel, voltage-gated sodium channel 1.6, which is clusters at the node, and they're in red. So when we compared the node length, so the length of the node from paranode to paranode, we actually found that there was a shift in the distribution of node lengths within the fimbria towards longer nodes. And when we averaged this per animal, we actually got a significant increase in the average node length in the hippocampal fimbria after spatial learning. So this is really interesting, but that doesn't actually tell us whether this is mediated by existing oligodendrocytes or whether this is just a consequence of the addition of new oligodendrocytes occurring. So I told you before that spatial learning induces oligodendrogenesis, or the formation of new oligodendrocytes in several regions involved in spatial learning. Well, that study didn't actually look at the fimbria, so we confirmed that we do get new oligodendrocyte addition in the fimbria after learning. So in order to determine whether these longer nodes were a consequence of just new internode formation and therefore less mature nodes are a little bit longer, or whether it is actually driven by pre-existing oligodendrocytes, we prevented the addition of oligodendrogenesis by deleting the MYRF gene from OPCs. So as Ben alluded to before, if we delete MYRF from OPCs, they can't actually differentiate and form into oligodendrocytes and form myelin. So we are preventing new myelin addition. When we did this and we trained the mice in the radial arm maze, we actually found that we still got this shift towards longer internodes in distribution and an average increase in the node length. And just to confirm things, we then I then measured the nodes that were flanked on either side by an existing oligodendrocyte. So these are the GFP labeled oligodendrocytes that were labeled prior to learning. So these internodes existed before any learning began. And when we measured the nodes that were flanked on both sides by a GFP internode, we saw that there was, again, a shift towards longer nodes, and didn't quite reach significance, but a tendency towards an increase in the average node length. So what could be driving this? Are we actually increasing the thickness of an individual sheath that could then be adding an extra myelin layer to encroach on the node? So to get at this question, we actually we did cross-sectional TEM in the hippocampal fimbria to look at, and we first measured the G ratio, which is a rough indication of myelin thickness. So it's the axon diameter to the fiber diameter ratio. So an increase in G ratios is indicative of thinner myelin, which was somewhat unexpected because we saw an increase in the G ratio, suggesting that the myelin around fimbria axons in the learning mice was actually a little bit thinner. When we looked a little bit more closely and actually quantified the myelin lamellae, which are shown here by these major dense lines, so each individual wrap, we actually saw there was absolutely no difference in the number of wraps. And that left me scratching my head for a little bit, but when I was quantifying this, I actually noticed that the space between the axon and the myelin sheath itself seemed to be slightly different. So I went back and re-quantified this, and I actually found that there was a decrease in the size of this space called the periaxonal space. So this is a fluid-filled space that is actually really important for modulating conduction velocity as well. So with the help of Professor Renaud Jolivet, who at the time was at the University of Geneva, but has just moved to Maastricht University, we actually wanted to know what these subtle ultrastructural changes might be doing to conduction velocity. So we modeled the fimbria axons computationally, and so this is using the experimental parameters the experimental measures that we gave, and we found that in the no learning mice we had a average conduction velocity modeled at just under 1 meter per second. When we changed the node length to that measured in the learning mice, just the node length itself, we got an increase in the simulated conduction speed. Same as when we changed the periaxonal space itself, which we've labeled here as altered myelin, we also got an increase. And when we added these two things together, we actually got this compound effect in simulated conduction velocity, and that models the learning parameters that we got. So we were expecting faster conduction along these fibers. I then went and actually measured this. And so we did compound action potentials in the fimbria fornix in horizontal ex vivo sections after learning or no learning. And so we found a roughly similar average conduction velocity to what we had simulated, so just under 1 meter per second in the myelinated fibers. So compound action potential, you get 2 peaks, and so the first peak is largely the myelinated axons, and then the second peak is largely the unmyelinated fraction of the axons. And so when we measured this in the learning mice, we actually saw that the compound action potential in the myelinated peak, the myelinated fraction, was significantly increased, so much faster, similar to that what we expected from the simulations. But there was actually no change whatsoever in the conduction velocity in the unmyelinated portion. So this suggests that it actually is certainly myelin-related changes rather than axon-related changes that are driving this change in conduction velocity. What was really interesting is when I plotted the measured conduction velocity against the improvement in the radial arm maze performance, so the reduction in the total errors from day 1 to day 11, I actually found that this was correlated quite well. So those that had faster conduction velocities tended to have a greater reduction in the radial arm maze errors over the training period. So it suggested that if we can increase conduction velocity somehow, then we may be able to learn a little bit better, or the mice learned a little bit better. Okay, so far, what I've shown you is that learning induces adaptive changes in the node Ranvier length and the periaxonal space width, and this is independent of oligodendrogenesis. So this is driven by the existing oligodendrocytes. And that these changes synergistically enhance conduction speed along the Ranvier axons. But this leaves us with a number of questions. So does this kind of plasticity occur in all types of learning? Or is it specific to spatial learning? Do mature oligodendrocytes become less plastic during aging? And what is the actual mechanism? What is driving this adaptive change? And I guess one of the other questions is this adaptive change actually really critical for the acquisition of spatial learning or other types of learning? What I'm going to do for the last very few minutes of my talk is give you a little bit of a sneak peek for what I've done so far to address this first question. Okay, so again, using the PLP DFP mice, one of the types of learning that I have done so far is contextual fear conditioning. We chose contextual fear conditioning because this is also hippocampal dependent, but it's a much shorter protocol and involves a lot of different brain regions within the acquisition of the fear learning. So the protocol that I have is fairly standard. So on day 1, they were habituated to context A, which is just a black plexiglass box. It had white noise playing in the background and the lighting was consistent. On day 2, mice were either conditioned or they were just exposed to the box again for 5 minutes. Those that got conditioned actually had 3 foot shocks spaced a minute apart during that 5 minutes. And then on day 3, I tested the fear memory from these. So they got exposed to both context A, which is the conditioned context, and a novel context, which was entirely different. It was clear plexiglass circle, had no grid on the bottom, very flat surface, and the lighting was slightly different. So just to show you that these mice actually were conditioned. So I'm showing you the data just from the animals that I have done the additional analysis with. And so you can see here that those that were conditioned actually spent more time freezing, or kind of greater proportion of time freezing in context A, and there was absolutely no difference in context B. So this shows that they did actually get conditioned to that context. And this is just showing you that generally freezing from the day 1 habituation, the amount of time freezing from day 1 habituation went up in those that were fear conditioned and actually went down in those that were not. So this is all in context A. As I mentioned before, contextual fear conditioning actually involves quite a lot of different brain regions, and it's been relatively well mapped out. But in the interest of time, I'm not going to talk a lot about it, but I am going to say that I'm looking at a very specific circuit, and that is the projection from the ventral hippocampus through the fimbria to the prelimbic cortex at the front, and the involvement of the basal amygdala. So just this kind of bottom left quadrant of the circle. So looking at nodes that were flanked on either side by a GFP internode, when I looked in the fimbria after contextual fear conditioning, there was absolutely no difference in the node length. There was no change in the distribution and no change in the average node length. But what was really interesting is that in the prelimbic cortex, we actually got this really subtle shift towards longer nodes.
And that didn't quite reach significance when we averaged it out per animal, but it was approaching, and we've still got a little bit of work to do. So this suggests that there's some regional specificity in the type of learning that's going on. And we also had a look in the basal amygdala, in the body of the basal amygdala. Sorry, I don't have the nodes, but I thought this was pretty, just to show the green myelin that you can see here. And we looked at nodes that were actually in the body of the amygdala here. And we saw no difference in the node length or the average node length in the basal amygdala. So I think I'm right on time, so I'm going to finish up there, and I need to thank a number of collaborators, specifically Carlie, our past PhD student who's now in the US, Renee Pepper, who was co-post author on the published work, Renaud, who did the computational modeling for us, and specifically John Harry, who's an undergraduate student who worked over summer and did a little bit of volunteering, who did all of the node analysis in the contextual feature. So I'll leave it